Hey, some intriguing new findings have come from a groundbreaking study in the UK where volunteers were deliberately infected with the SARS-CoV-2 virus to understand why some people naturally avoid getting COVID-19. This research, led by University College London, UCL, is the first to track the course of the disease from the moment the virus enters a person's nose to the development of illness, opening the door to better vaccines and treatments. The study, called the UK COVID-19 Human Challenge, involved 36 healthy young adults who volunteered to have the virus introduced into their noses. Before participating, all volunteers were screened to ensure they were at low risk for severe disease and had no significant comorbidities. The researchers used the lowest possible dose of the virus that could still cause infection. Shobhana Balasingham, a research lead in Wellcome's infectious disease team, emphasized the value of human challenge models in understanding how the body responds to infectious diseases. These studies allow for close monitoring of the immune response from the moment of infection through to the development and severity of symptoms. After infecting the participants, the researchers monitored the cells in their blood and nasal linings immediately to see what happens right after exposure. This detailed analysis resulted in a dataset of over 600,000 individual cells, which are now part of an effort to create a comprehensive reference map of all human cells, known as the Human Cell Atlas. In all participants, the researchers found that specialized mucosal immune cells in the blood were activated and there was a reduction in inflammatory white blood cells, which typically surround and destroy pathogens. However, in participants who either resisted infection or tested positive but avoided a full systemic infection, the researchers observed a unique immune response. Specifically, they found high background activation of a gene called HLA-DQA2, which signals the presence of a viral invader to other cells. Additionally, those who best resisted the virus showed a quicker immune response in specialized nasal cells and a slower response in blood cells compared to those who became infected. Participants who contracted COVID-19 only mounted a nasal response after 5 days of viral exposure, indicating that activation of nasal cells is crucial for fighting this and potentially other coronaviruses. The researchers also discovered recurring patterns in amino acids among activated T-cell receptors, which are cells that target and eliminate infected cells in the body. These amino acid patterns, known as motifs, suggest a shared ability to recognize something in the SARS-CoV-2 virus. Emily Mobley from the Wellcome Sanger Institute highlighted that identifying these motifs could lead to engineering virus-specific T-cells for treatment, not just for COVID-19 but for a range of diseases involving immune attack. Marko Nikolic, the senior author of the study at UCL, expressed hope that their research will lead to better ways to combat pathogens. He stated that these findings provide a much greater understanding of the full range of immune responses, which could form the basis for developing treatments and vaccines that mimic these natural protective responses.